Hi everyone, thanks again for attending our webinar today. I'm very excited to introduce you to the BioFlux system and to talk about how it can be used for cancer biology assays. Today we're going to start with a discussion of some of the parameters that are important for cell biology assays. We'll then explain the BioFlux system and how it works, and we'll look at some of the applications of the system for cancer biology and related biological applications. And finally, we'll finish up with what a BioFlux system looks like and how it's configured. What BioFlux offers and what we're going to talk about is an added dimension or added dimensions of control over several critical parameters that influence biology and that are not all controlled for in a single live cell adhesion or Boyden chamber assay. Some of the controllable variables delivered by the BioFlux are shear, which we'll discuss in the following slides, maintenance of consistent conditions, for example, for compound delivery, environmental conditions like temperature and atmosphere, as well as the ability to perform these in parallel and to collect dynamic data in time-lapse, especially important for the assays we'll talk about today. All of these parameters can potentially enhance your understanding of the dynamic events occurring during the metastatic cascade. There are two ways of thinking about fluid flow in the BioFlux. One is that the flow delivers shear, which is a component of the in vivo niche, and two, that flow can be used to create dynamic conditions within a reaction container to enable novel assays. Here, for cancer biology, we can think of the spread of cancer cells during the process of metastasis, which includes processes like cell adhesion to the endothelium and transmigration of the cells to the underlying tissue, which fall into the category of physiological conditions. And then we think about dynamic events like the actual process of a cell sensing a chemokine and moving actively towards a target space, something that you normally look at in a Boyden chamber, and we can use flow plus or minus shear to study these events. The first parameter we're going to talk about is shear. Shear is a mechanical force. In this case, we're talking about something that's exacted by fluid flow or blood flow, where in a healthy individual in the circulatory system, the flow can range, or the shear can range from less than 1 dyne to 20 dynes per centimeter squared. In the case of stenosed artery or other vascular disease, shear can be well in excess of 20 to hundreds of dynes per centimeter squared. This ever-present force has a large influence on biological processes, causing changes in gene expression, leading to good outcomes like we see during embryonic development, and pathological changes leading to blood clot formation and inflammation. So it makes sense that shear should be a controlled parameter in many experiments, especially those in cancer biology, stem cell biology, the cardiovascular niche, and inflammation. Here's a good example of where shear plays a very important role in vivo. The biology of the endothelium is always under blood flow. You can't separate the biology from the actual mechanical forces. When endothelial cells are grown under shear flow, there are changes in cell signaling leading to morphological and functional changes in cell behavior. Specifically, in the case of cancer biology, it's known that shear applied to endothelial cells can either increase or decrease, depending on the type of cancer, the propensity of cells to adhere to the endothelium, which in turn influences how likely it is for a cancer cell to be found in a secondary lesion. It also influences how well cancer cells interact with the extracellular matrix. Just to finish up talking about other important controllable parameters besides shear stress, if you include flow in biological assays, this confers a number of additional benefits, such as full management of the microenvironment that the cells see, which is important to ensure consistent cell behavior time after time. As well, this provides the opportunity to change conditions reliably for other types of experiments, for example, to create areas of continuous increased chemoattractance to enable chemotaxis, and to provide pulsatile flow to understand the role of cyclic waveforms on cell biology. How does the BioFlux deliver these controllable parameters? Well, it was designed exactly with multi-parametric microenvironmental control in mind. Using the hardware, which we'll talk about shortly, and the microfluidic BioFlux plates, we can tightly control parameters of shear from 0 to 200 dynes per centimeter squared, temperature, ambient to physiological, use of external gases from 5% CO2 to 100% nitrogen to buffer media or provide anoxic conditions for hypoxia models. Our unique fluid control interface allows the channels to be compartmentalized so that assays can be run under two conditions at once. All of the above benefit live cell assays. In addition to controllable environmental parameters, the BioFlux was designed to be easy to use. The consumables are in a well plate format, which we'll talk about in a minute, so many assays can be run in parallel, and it's easy to clean up and set up. And because we use a microfluidic format, we use smaller amounts of reagents, and this opens up the possibility of using precious clinical samples and rare cells to their full potential in cancer biology assays. At the heart of the BioFlux system is the pneumatic controller, which affords very tight control of fluid handling and shear, and that's shown on the upper left. The controller couples to the well plate microfluidic device, which is shown on the upper right, using the airtight interface, which you can see next to the controller on the upper left. Then data can be collected using microscopy, either manual or automated, so you can see that on the lower left. And user-friendly software enables both seamless data collection with concurrent flow protocols and automated data analysis. So you can see a screenshot of that on the lower right. Each channel of the well plate microfluidic is, for all intents and purposes, a flow cell, but it doesn't have the baggage that's associated with a normal flow cell or the fluid volumes associated. The BioFlux plate serves as a self-contained array of flow cells. Here are some of the features that make the BioFlux plate itself conducive to live cell assays. Number one, the well plate format makes for easy lab handling. Two, the cover glass, which is on the bottom of the plate, is optimized for imaging. Three, channels are identical to each other for experimental repeatability. And four, the design really favors simplicity. There's no liquids in tubes, so you don't have any contamination. The experiments are self-contained within the plate, and there's no setup overhead or cleanup overhead. Okay, let's talk about the BioFlux well plate microfluidic devices. This is really at the heart of the system. They're currently configured in two main formats, 24-well and 48-well, with slightly different uses for each. The 24-well design shown here was engineered for experimental flexibility. There are 8 channels, and each channel has 2 inlets and 1 outlet. This enables compound switching on the fly as well as compartmentalization of the channels using parallel flow, which we'll talk about later in this presentation. This design is currently intended for physiological shear. The 48-well plates come in 2 shear ranges, enabling both physiological shear, or lower flow rates, and pathological shear, or higher flow rates. Each channel is formed by 2 wells, 1 input well and 1 output. This enables higher points. See the channel design right and how it is arrayed over the plate on the lower left panel. So how does the BioFlux work? It's very simple. The walls of the plate serve as below. So you fill the wells with whatever you're interested in running through the channels, either cells, buffer, medium, and then you place the cover or interface on the top, which forms an airtight seal. The controller then delivers pressure to the well, which pushes the fluid through to the well on the other side. The experiment can then be observed using an inverted microscope. And as I said before, the bottom of the plate is formed by a number 1.5 cover slip glass, which is really optimized for imaging using the inverted scope. Okay, so what do we use the BioFlux for? That's probably what you're most interested in hearing about. BioFlux applications run the gamut from the cardiovascular space to microbiology. Today, as you know, we're going to focus on those assays that are relevant to cancer biology, such as endothelial cell biology, rolling, cell adhesion, transmigration, and chemotactic migration and invasion. Please let us know if you have any questions about assays we haven't noted here or assays you would be interested in learning more about using the chat box. Okay, to start, we're going to focus on applications that are relevant to shear or the shear delivery aspect of the BioFlux controller. So we're going to start with cell adhesion. This is what's occurring within the vascular niche in response to conditions at the endothelial wall. So this includes shear stress, as I mentioned. Here are several stages of cell adhesion in blood vessels. You have things ranging from no interaction, transient interactions, where the cells make small, short contacts and then continue to roll along the substrate. and then that transitions to actual rolling, where the cells are truly contacting the substrate continuously, and you can observe this as a reduction in velocity. And then finally, real cell adhesion, which can later lead to things like transmigration and other phenomena. And so you can observe all of these in the BioFlux. So first we'll talk about capturing dynamic data for cell rolling. This is really the first step of cell adhesion. What you can see at the top of the slide is a stacked micrograph over 30 time points where the cells that appear as white circles are rolling on the substrate, which in this case is VCAM1. The distance versus time can be measured to determine the velocity using the BioFlux software. This can be used to characterize receptor-ligand interactions, response to inhibitors, or other variables like different cell types, mutations, and that sort of thing. The BioFlux can be used to study adhesion to purified ligand or to cellular monolayers. Shown here on the top are data from both types of experiments. In one case on the left, cells are added to purified ligand under different flow conditions, and in this case we also have different conditions of inhibitors, and then the cell number is counted during the experiment. In the case on the right, cells are added to stimulated and unstimulated monolayers of endothelial cells, so this is an example of a cell-cell adhesion assay, where then after that the adhesion is quantitated. On the far right at the top, you'll actually see an adhesion assay for colon carcinoma cells adhering to endothelial cells. And you can see that there's a differential effect based on whether the endothelial cells were activated with a chemokine or resting. In the case of the colon cancer cells, they preferred to adhere to resting cells. So that's very interesting. The BioFlux can also be used for higher throughput studies of cell ligand or cell-cell adhesion under flow to the point of creating compound response data like IC50 curves. Shown here on the bottom left are two panels taken from a white blood cell adhesion assay on VCAM, which could just as easily be a tumor cell adhesion assay under flow, where the top channel is a no drug control and the second channel below it is a treatment with a high dose of anti-VLA4, which blocks VCAM adhesion. Shown on the right are IC50 data for 3 anti-VLA compounds. What is very interesting about capturing data using microscopy for cell adhesion is that unlike using a plate reader, you collect more data than just the raw fluorescence or the bulk fluorescence. In this case, 2 inhibitors against the same target are used, which would score exactly the same in a plate reader assay on the left panels. However, here you can see that something funny is going on with the panel at the top, a potentially undesirable outcome of the adhesion inhibitor which is cellular aggregation. This is depicted on the right by graphing the cellular area against the shape. For normal cells, the area is similar and the shape is round, which you see on the bottom right panel. For the clumped cells, the area and the shape is irregular, and because you have these microscopy data, you can eliminate this compound from your screen right away. Okay. Moving along to the ultimate outcome of cell adhesion, here is an example of a vertical transmigration assay done in the BioFlux channels. The assay is set up by growing a confluent monolayer of endothelial cells in the BioFlux channels. Once the cells are confluent, they can be stimulated, for example with chemokines and or shear, and then circulating cells, such as tumor cells or white blood cells, can be introduced into the channels under shear flow, where their behavior can be observed. In this case, a time lapse is shown where a single PBMC is actively transmigrating from the top of the monolayer to the bottom of the monolayer, as evidenced by a change in both shape and contrast. So you can see the cell goes from very phase bright to very phase dark over the course of the time lapse. Transmigration can also be performed as an endpoint assay, an example of which is shown here. Cells are flowed into the channel as if this was an adhesion assay, which you can see on the bottom left panel, and then the temperature is increased to physiological or 37 degrees, and this is permissive for transmigration. So then you can see on the bottom right-hand side, cells that have transmigrated can be enumerated following the shifts. You can see all the phase dark and amoeboid-like cells that have transmigrated. Now let's switch gears from talking about assays that heavily rely on shear to assays that we can accomplish very well in the BioFlux using flow. So we're going to start out by talking about cell motility, cell migration, and invasion. Many cell types are screened for motility phenotypes using Boyden chamber assays or transwell inserts. This provides a static data point in an otherwise dynamic process. The BioFlux, in contrast, can be used to enable a dynamic recording of these data using parallel flow. This enables the entire process to be screened in the channel, and this includes applications such as migration, invasion, and endothelial cell development. In a way, you can think of the BioFlux channel as a lateral Boyden chamber or two-compartment assay. So what do I mean by this? A specialized use case of the BioFlux exploits the laminar fluidic conditions found within the channels. Using the 24-well device, flow can either be intermittent, meaning we're flowing from one inlet or the other, or in parallel, meaning the two inlets are running at once, and this is what we're talking about now. Due to the fluid dynamic conditions within the channel, the two fluids remain compartmentalized in discrete sides of the channel. This is what you can see on the bottom panel. There are two different types of fluorescent beads flowing in parallel, and you can see that there's no mixing, so you see the red and the green side by side. In the case of assays in the BioFlux system, we can use the parallel flow for two fluid compartments to study things like chemotaxis, or we can partition the channel into a liquid and a solid zone for invasion studies. For invasion assays, we can use parallel flow to create a gel region inside the channel as if you were using a Boyden chamber turned on its side. The gel can be dosed with compounds or growth factors, and cellular response can be followed in time-lapse. What you see here are channels that have matrigel in half of the channel and the other half is still patent. On the right is a channel with large beads trapped inside the gel, which is the bottom part of the channel, and small fluorescent beads that are flowing in the unobstructed side of the channel. Special cases of cell motility in response to stimuli. All phenomena are important in health and disease, and each step of each of these processes can be targeted for drug development. Therefore, understanding the temporal events during these processes is very crucial. So let's start by talking about chemotaxis. The phenomenon of chemotaxis can be thought of as a response to a call to action by a cell. This example here shows the normal response to an injury by a neutrophil, where it's attracted by a chemical message to go where it's needed to perform its duty. We can study this activity in the BioFlux. Shown here is a chemotaxis assay where we are using neutrophilic-like cells within the channel, and then we flow FMLP, which is a very potent chemokine, in from one of the inlets and buffer from the other inlet using parallel flow that I explained in the previous slides. We then collect time-lapse data of the cellular response over an hour. Each track on the upper left, you can see in color, is actually a path one cell took over the time-lapse. And then we're able to use this to get dose response to the chemokine, which is shown on the top right panel, where the bright orange is the number of cells in the chemokine-containing compartment at the end of the experiment. As well, if you look on the bottom where those black charts are shown, we can follow other parameters like velocity and movement trajectory using software called multidimensional motion analysis. Moving on from chemotaxis to invasion assays, so this is where we're using a solid in the channel versus two liquids. We'll talk about angiogenesis. Angiogenesis is a multi-step process that starts out relatively simply with a single cell, to be exact. What happens is a single cell gets a chemical message and then responds to that message by starting the process of angiogenesis. And in the case of cancer biology, this contributes to the vascularization of tumors. So on the top right, you can see a highly vascularized tumor as a result of the process of angiogenesis. The BioFlux has an important advantage in the study of the very early steps of angiogenesis, namely the matrix degradation and invasion steps, where it adds the ability to capture time-lapse data, use physiological shear, or add and subtract growth factors. In addition to flow, in this case, shear is very important and has been studied in the literature using collagen plugs in parallel plate flow chambers. Shown on the right is just that, endothelial cell invasion and sprouting with and without shear. Below left is a cartoon of some of the responses that are seen when endothelial cells are exposed to angiogenic growth factors such as VEGF. and you see things like sprouting of the cells and branching, etc. In the BioFlux system, we use Matrigel in one half of the channel and continue to flow media over endothelial cells that were present in the channels under starvation conditions. Within about 2 hours, in this case, endothelial cells will degrade and invade into the gel. And in this particular experiment, we added an angiogenesis inhibitor to the gel on the bottom right and found that the cells could attach to the gel but could not enter and appeared to be lined up along the interface. So you see that on the very bottom right panel. We have also observed in time-lapse cell sprouting and apoptosis in these cells. Likewise, metastasis involves movement of cancer cells from the primary tumor to other sites of the body. Again, this is a dynamic multi-step process which can be investigated in the BioFlux system using parallel flow and the invasion capability of the system. So shown here are the time-lapse data for 15 hours of time-lapse for invasive HT1080 colon carcinoma cells, which are the panels on the left, and another cell line, which is a less invasive breast cancer cell line called MCF7 on the right. What you can see in these panels are, each panel is 1 hour, and you see the channels, which are the straight black borders, and then you see an area that is totally devoid of cells, and that's where the gel is deposited in the channel. And then you can obviously see the cells on the top. And both sides are the same, except for the cell type. So starting around 4 hours, where you see the purple arrows, you can see that the HT1080 cells are beginning to invade the matrix. And they invade the matrix vigorously over time. And that if you look at the MCF7 cells and you look at the gel partition of the channel, you can never see any invasion. Other similar experiments can be performed in the BioFlux to characterize the invasiveness of other different cell lines and the temporal response to inhibitors or even the response to unknown compounds. Shown here is the measurement of cell invasiveness over time from the previous panels where the dark orange line shows the amount of gel occupied by the HT1080 cells, which we saw in the previous slide, and the light orange shows the comparative cell line, MCF7, which cannot invade the matrix, and that's the line that goes along the x-axis. So that concludes my discussion of applications. I just want to summarize and say that I hope during this brief webinar you've learned that shear and flow can be used to benefit and enhance your cancer cell assays. I also hope you've learned a little bit about the BioFlux and how it provides control and ease of use to live cell assays, including physiological parameters to mimic the vascular niche and dynamic data capture, which is very important for assays for chemotaxis and invasion. And finally, how you can use the one BioFlux system to perform many types of assays. Here are the two product offerings for the BioFlux. The 200 system, which is shown at the top right, consists of the controller, software, and accessories, which is everything that you need to run your assays if you have your own inverted microscope. And the BioFlux 1000 and 1000Z that's shown on the bottom, and those are both fully automated and integrated systems that include an automated image capture workstation and all the hardware and software that's needed to run walk-away automation of these assays. If you have further interest in the system, would like more information about the system itself or applications, please visit fluxionbio.com. Or to inquire about ordering, please contact our sales department at sales@fluxionbio or at 866-266-8380. Thank you again very much for attending our webinar this morning.